I'm Reza, and I'm going to talk about functional brain imaging today. It's a relatively short overview of the functional brain imaging that we do at the Brain Research Institute. So at the Brain Research Institute, we do two different types of functional brain imaging. Kind of like one is functional magnetic resonance imaging, or fMRI, which participants go into the MRI scanner, and actually I can take this out. So participants go into the scanner. It's a very large magnet, and it measures the blood oxygen level, and so it's an indirect measure of neural activity. The other type is electroencephalogram, or EEG, that we do it at the Brain Institute. So basically, we put some electrodes on the scalp, and we record electrical activity of neurons firing. And so basically, a population of neurons that firing, we record that electrical activity on the scalp. So comparing the two, the temporal resolution, or how fast we can see changes, is much better in EEG compared to fMRI, but fMRI can look deeper in the brain. So for the rest of the talk, I'm just going to focus on the EEG, and fMRI is mostly Tracy Melzer's expertise. So here, I'm going to show you a recording of EEG that we did with Robin Webster. She's an artist, and we recorded her EEG while she was weaving. And so you can see at the bottom that the electrical activity is being recorded, and it's kind of like some wiggly lines coming through, making sense of it slightly on the top one. So I've back projected it onto the scalp, and so you can actually see what regions of the brain are kind of lighting up. And so looking at those lines, and kind of like very complicated and very meaningless just by looking at them, what do we actually expect to see in there? So there are slow waves, to fast waves, so how quickly it's changing. For instance, when we are sleeping, we expect to see very slow waves that are dominant, and when we are learning, it becomes basically the fastest. So based on the speed of the activity, we can identify what's happening underneath and what type of processes are happening. So basically, when we see a new signal, we can break it down into the slow to fast components of it and analyze it that way. The other approach that we can look at EEG is by asking a participant to repeat doing a task over and over again. And the reason for that is it's kind of like a consistent pattern keeps happening every time that we do a task. However, there are so many other things happening at the same time that on a one time that someone does something, we can't actually identify that consistent pattern. But if they do it enough times, then over many times, we would start seeing a consistent pattern, as you can see, kind of like in the middle figure there. So on the right, we ask participants of the Parkinson's study to basically click if they see a type of stimulus, and ignore it if they were other types of stimuli. And so we can actually see that looking at Parkinson's and control participants, we see a relatively similar pattern. And by looking at that, one could identify speed of processing, amount of attention, complexity of the task, and so on and so forth. So as I mentioned earlier, one of the problems with the EEG is identification of activity inside the brain. So what we do here is we look at a structural image, which is someone's MRI. In this case, that's mine. And then we do some math, and we create a mathematical model, which is a 3D representation of that. And on the three-dimensional one, I have put the electrodes kind of superimposed on that. So you can actually see when we put the electrode cap on individuals, the electrodes, we can actually map them back onto the 3D model. And so based on that 3D model and the activity that we've recorded, we can then do an inverse and identify what regions of the brain, what parts were more active. And so one could ask, so why that's important? For example, let's look here. So here is we are looking at the components of activity while a person was sitting, having their eyes shut, and just being awake and rested. And so in that moment, we would expect to see that kind of like elevated peak around alpha activity, which is happening there. So then we look at a few of the individuals. So across the groups, we can see that, yes, in controls, it is happening in the posterior occipital regions, and that starts declining as we go towards dementia. And so that's interesting, but we can go one step further and we can map that into the actual brain to see what's happening inside the brain. And so looking at that across different speeds of activity, we can see that the slow waves seem to increase as the disease severity increases, and the faster waves seem to decline with the progression of the disease. Another kind of interesting thing that we can get out of the signals is how they move together and how they work together. So on the left, I've generated signals that you can see that the first two, we could agree that by knowing that one is going up or down, we could say the other one is doing down or up. However, between the first one or the second one and the third one, we can't say that anymore because that relationship has faded. And so we can say that the first and second one are functionally connected and the other ones aren't connected. So what can we do with that? We can actually look inside the brain and see what happens between the synchrony of different regions of the brain as disease progresses. And we can look at slow waves, the fast waves, and we can see that there are some changes that are associated with progression of the disease. So the next step that I'm trying to do is basically to identify, can we use those information on an individual level? So basically identify if we can understand someone's state of the disease, and potentially in the future, can we actually identify the trajectory of the disease that they are going to have based on the EEG measures that we have. I'm also looking to see, can we understand kind of more complex interactions that are happening inside the brain as part of that? So I guess that's my presentation for today, but feel free to ask questions afterwards. Thank you.